session, which is Dr. Mark Wirth. He is a material scientist here at EMSL, and he will talk about the application of machine learning for the identification of protein signatures from APT data. So Mark, please go ahead and share your screen.
Okay, hi everybody, thank you for joining me. I'd like to talk about our work that we've been doing here at EMSL to try and apply machine learning to identify protein signatures in atom probe tomography data. This is based on previously published APT data of ferritin embedded in resin. It seemed like we had all this data and we wanted to use machine learning to try and gain some additional insight into this data. APT is a technique that creates 3D point clouds of ionized atoms and small molecules. We are hoping that it may provide a complementary technique for determining protein structures. One of the problems with APT in this application space is that it provides no bonding information. So you end up with a 3D point cloud and you know where everything is in 3D space, but we don't know how everything necessarily is bonded. What we're trying to do is use machine learning to bridge that gap between 3D point space and actual structure. So I presume the first question on a lot of people's minds is what the heck is APT? APT is a technique in which the sample is sharpened to a needle, a sharp needle with a tip radius typically less than 100 nanometers. And this is, we apply high electric fields to the needle and pulse it with a laser. And every time the laser pulses, it sets off a time of flight mass spectrometer. And so ions are field desorbed from the tip of the needle. They go through this mass spectrometer and then they hit a position sensitive detector. The position sensitive detector allows us to extrapolate the position on the needle from which the ion came. And the sequence of events allows us to know where they live in the Z dimension, so as you're moving down the axis of the needle. And the time of flight mass spectrometer lets us know isotopically resolved masses of all of the ions that come off. So we end up with this isotopically resolved mass spectra, which we can create cool composition profiles and these 3D composition maps, which are interesting insights into materials. So the work that had previously been done by Danny Perea and others was they embedded ferritin in resin and they ran that through an APT. And the reason that they specifically chose ferritin is that the protein embedded in resin is difficult. It's difficult to decouple the signature of protein from resin. But if you have an iron core like ferritin does, it provides this great fiducial marker for exactly where your protein should be. So to get this into the atom probe, the ferritin had to be suspended in liquid resin, which was cured, and then we did a fib-sem liftout. The resin used was low acryl, so there is which has no nitrogen-containing compounds, allowing us to look at the protein and the resin and discern the actual protein from the resin based on where there is and is not nitrogen. The way that we prepare samples in the atom probe is we use a fib to do a lift out from a bulk sample. And this lift out is typically on the order of 10 to 30 microns. And then we take this and attach it to a silicon post. These silicon posts are truncated cones, like a waffle cone. And we take the lift out and we chop off chunks of them and we attach them to silicon posts. These silicon posts are highly conductive and just make a great substrate for generic atom probe running. And then we use annular milling in the fib to actually sharpen these down to needle shapes. So if you look in the bottom middle figure, you can see an actual ferritin tip, one of the ones that we actually ran. The top part is ferritin and resin, and then there's platinum, which we use to weld to the silicon micropost. Typically, we produce anywhere from 1 to a dozen samples per liftout, usually more around the range of 4 to 6. So we're trying to get from APT data somehow to produce protein structure. So this is our like big lofty goal right here. So on the left you see ferritin core, so the iron core is the green, and then around it there are CO and CnH2 ions. And then on the right is an actual ferritin protein just downloaded from Protein Data Bank. So how do we get from one place to the other? Well, we're trying to use machine learning. And then our machine learning workflow is in order to use machine learning for any task, as most of you probably know, you need lots and lots of training data. So if we want to produce some sort of model that can look at data and say this is protein, this is not protein, or say more about it than that, like this is the structure of the protein, this is what it looks like, you need to feed it lots and lots of data so that the model can learn how to label those things. So to get there, you need labeled data, which is nice in dealing with proteins because there's the Protein Data Bank. So the Protein Data Bank is this database, as I'm sure a lot of you know, of protein structures, which is freely available online. And since machine learning tends to be so data greedy and the data has to be labeled, it's an excellent resource for us. But one of the problems with, we can't directly apply PDB data to atom probe data specifically because they're not exactly the same. So PDB data is this atomic data, like it's actual protein, there's bonding structure, you know everything about it. But our APT data is fragmented into mass spectra. So we have CN ions, CO, CnH2s, all these things, as opposed to just atomic resolution. So we needed to artificially fragment the PDB data. And our detector efficiency on the atom probe is only about 40%. So we had to remove 60% of the data from the training data. And there are stretching and compression artifacts associated with reconstructing atom probe data. Usually tends to be between about 0.9 and 1.1. So we simulated that in the training data. And then to simulate uncertainty in the training data, we added a Gaussian distribution to every atom and molecule in the data. The uncertainty in the atom probe is on the order of about 0.3 nanometers. And we also input some experimental APT data so that the model would actually know what APT data looks like. And then from there, we had to label the data. And our labels we chose were whether they were or not part of the protein backbone. We chose that because our ultimate goal is to try and say something more than just whether or not protein is present. So this first step is just to say if we can or cannot determine whether something even belongs to the backbone. And then you cannot directly feed 3D point clouds into a neural network. And so the approach we chose was using nearest neighbors. So the nearest neighbors for every single atom and molecule in the data set were calculated, and the distances to those neighbors and the identities of those neighbors were what actually went into the neural network as training data. And there's a schematic of our handy neural network. As a control experiment, in the previous work with the ferritin, one of the data sets that was run was hematite embedded in resin. So hematite is similar to ferritin in that these iron nanoparticles simulate the ferritin core. So what we wanted to do was apply the neural network to this data set and see what came out. Ideally, when you're applying it to this, it should see nothing at all. There should be no protein because there is no protein. So when we applied it in about just under 6 million ion data set, there were fewer than 100 false positives. So we were pretty happy with that result. And the next step was to actually apply it to ferritin and resin. So what you see here is a 3D point cloud of ferritin in resin, which is, if you look at the blue, those are the iron cores, and the red is the resin. And the green here is what the neural network labeled as protein backbone. So the neural network had access to the whole data set, and it labeled all it labeled was this is or is not protein backbone. Everything that was left over, we extracted the iron containing cores because we knew, because anything containing iron, you can just extract that directly. And everything that was left over was assumed to be resin. So if you look on the right, this blue are the iron cores, and the green is the protein backbone. So the protein backbone definitely seems to be surrounding a lot of the iron cores, which is exactly the result that we would be looking for. One of the things that we do to quantify a quality like this, because we're just qualitatively looking at this and say, hey, it looks like there's protein around that core, but it's just what I'm looking at. So one of the more quantitative methods we use in APT frequently is what we call a proxygram. So a proxygram, the blue is the concentration of the iron cores, and the red is the concentration of resin. So a proxygram works in that if you imagine you're at the surface of one of those iron cores from the previous slide, if you move out away from the surface, you should be increasing concentration of resin and decreasing concentration of iron ions. So if you're moving to the left in this plot, that's exactly what's happening. The resin concentration is increasing, the iron concentration is decreasing. And then as you move into the core, you should see the concentration of the resin decreasing and the concentration of the iron increasing, which is exactly what you see. And then what you want to see with this protein backbone is we want to see somewhere near that interface, there should be a peak of protein backbone, and then it shouldn't really exist very much anywhere else, which is exactly what we found. So there was a nice peak just near the interface of where the protein backbone actually exists. So we found that our neural network could successfully identify the protein fragments in resin, which is exactly what we were going for. In the future, though, what we really would like to move towards is something more like semantic segmentation. That's our big lofty dream goal. So semantic segmentation would be we can actually run atom probe data with proteins in it and actually segment the proteins and know information about them. So identities, structure, etc. So to achieve that goal, we are poking several different methods. One of them is tracing the backbone. So if you look on the right, that image is some of our first attempts at actually trying to understand how to trace backbones. So that is actually PDB data, but we use an algorithm in which you move through the protein trying to minimize the distance traveled. It's difficult to see specifically in this jagged data set, but we do see some actual structure there. So that's one of the ways that we can work towards. We were not using convolutional neural networks in this work. It was just more computationally expensive than we really had access to. So in the future, using 3D convolutional neural networks could definitely be a direction that we go. And then we are also currently working on analyzing proteins in water ice. So water ice presents a whole different slew of challenges, but doing cryo experiments like this, we are perfectly capable of doing at EMSL. But if we can actually use water ice instead of resin, we can focus more on applying the machine learning to try and discern something more about structure and identify more information as opposed to simply decoupling resin and protein and just like, hey, this looks like there's protein here and this looks like resin, but we can actually hopefully move past that and say more interesting information about it. And then also we are limited by detector efficiency. So our atom probe only, as I mentioned, our detector efficiency is about 40%. The newest generation of atom probes can be up to 80%. So that would make a significant difference in our work. So if we can find a way to access data or run data through a newer generation of atom probe, then I feel like that would open up a lot of potential information in this algorithm. So thank you to everybody. Thank you for attending my talk, and thanks to EMSL Capability Development for funding us, and thanks to Danny Perea for helping so much. And that's it.